This is bio367 lecture component for the topic of prokaryotic genetics. This specific part of the overall lecture will focus on the processes of conjugation. Conjugation is often referred to by bacterial geneticists as bacterial mating, and it's a physical process wherein two cells will physically attach to one another through the formation of a sex pilus, and that's what's shown in this little cartoon here. An F plus cell will conjugate with an F minus cell, or an Hfr cell will conjugate with an F minus cell. In either case, cells that contain something called the F factor or fertility factor will be able to detect those who don't. They are referred to as F minus, and they will move towards them and they will build a sex pilus, and then that sex pilus will serve as the physical structure through which DNA travels from the F plus or the Hfr cell and into the F minus cell. The F factor or fertility factor is a set or suite of genes. In total, it's about 40 genes, and included in those 40 are a set that are often referred to as the TRA locus, TRA for transfer. Okay? And within that TRA locus, which is within the F factor, are the genes for pillin. So pillin is a particular protein, all right, and it's used to make the structure referred to as the sex pilus, and also genes that encode proteins that are important, just generally speaking, in transferring and replicating the F plasmid. Those are genes for endonucleases, which are able to nick a single strand of DNA, and they're genes that play roles in replication of the F plasmid or a chromosome that has the F factor on it. So cells that are referred to as F plus have the F factor, and those that are referred to as F minus do not. Hfr cells have the F factor, and we'll talk about those in a moment, okay? So during simple conjugation, you have an F plus cell mating with an F minus cell. F plus cells have the F factor in a plasmid form, okay? So they have plasmid or F plasmid. This cartoon shows most of the steps of simple conjugation. So over on the left, if we start on the left, there's the F plus cell, okay? And here's the F factor, and it's found as the little green plasmid shown there in the figure. And the red is showing the rest of the chromosome. The F plus cell, a moment before this cartoon was being drawn, was able to detect the F minus cell. And when it did that, that initiated the expression of the genes on the F factor, which led to the accumulation of pillin protein and other proteins, and therefore the construction of the sex or conjugation pilus shown here. Okay, that's this structure right here. Once that has formed, then something called rolling circle replication begins. And in rolling circle replication, one of the two strands of double-stranded DNA that make up the F factor or the F plasmid will be nicked, and it will start to unroll and move into the F minus cell. That's what's happening in this middle portion of the cartoon. So as that happens, this right here is a single strand of DNA, okay? Now, as it moves into the F minus cell, then what you will have is replication to get back to double-stranded DNA in both the donor F plus cell and the newly formed F plus cell. So both of these are double-stranded DNA, okay? They are the F plasmid. When all of this is done, instead of having an F plus and an F minus cell, as you did over here in the beginning, you have the original F plus cell, which is still an F plus cell, and you've got the new F plus cell. Both now contain the fertility or F plasmid. For a video representation of that process, you can click on the link shown here. Now, I had mentioned at the beginning of this portion of the lecture that there were processes of conjugation. And in that, this is the second of two processes. So this is not simple. Instead, it is Hfr conjugation. Hfr stands for high frequency of recombination. In this mating process, an Hfr cell, which is shown over here on the left, is going to mate with a recipient or accepting F- cell, shown here on the right. Okay, so in an Hfr cell, the F factor is integrated into the chromosome itself. So you can see it there as green again, integrated into the overall red chromosome structure of the Hfr cell. And the genes still work the same. The Hfr cell is able to sense when there's an F- cell in the environment, and then it will start expressing the genes that are found in the F factor, which is shown in green. So it'll start building pillin protein. It will make a sex pilus, which is shown here. Okay? And once the sex pilus has formed, we will have transfer of genetic material from the Hfr cell into the F- cell. And what will happen is that transfer process will begin at a very specific location, almost on the very end of the F factor. And so as that DNA starts spooling in, and just like in the previous process, it will come in as a single-stranded molecule. As it starts spooling in, what will happen is primarily the red chromosome that carries the routine genetic information of the Hfr cell is what will make it into the recipient F- cell. If the two cells remain attached, physically attached for long enough, and that process can be about 100 minutes long in E. coli, okay? And there would be, strain-specific differences there, of course, but we know it's about 100 minutes in E. coli. Anyway, if those two cells stay attached long enough, then the entire chromosome, including the F factor from the Hfr cell, can make it into the recipient F- cell. Okay? And once it does that, what will happen is the single-stranded DNA that has entered the F- cell will align itself with homologous regions on the F- cell, and recombination can occur. So you can have substantial genetic change, and we talked about recombination in an earlier portion of this lecture. So if together long enough, the entire chromosome, including the F factor, can make it into this cell. Okay. Now over here you'll notice that this just says recombined F- cell. It doesn't say recombined Hfr cell. And the truth is that it is very rare for the F factor to make it into the recipient cell. Usually before that can happen, because note where the F factor is, most of it is over on the spot that would spool in last. Okay? Most of it typically does not make it in. So the recipient cell usually gains the possibility of new alleles because the chromosome, the routine chromosome, spools into the F- cell, but it rarely gains the F factor itself. And so it's usually the outcome of this is a recombined or a genetically different F- cell, not an Hfr cell. And that just has to do with time. That structure, that sex pilus will break apart before the F factor has time to make it in. Now it turns out that the fact that you need to have this physical contact in order for mating to occur was something that bacterial geneticists figured out quite a long time ago. And they figured out that this was a way that you could actually map bacterial chromosomes. And so very purposefully, bacterial geneticists designed experiments where they would mate an Hfr set of cells with an F- set of cells. And they would specifically choose strains of bacteria that had very different and detectably different alleles of specific genes that could be screened for. So in this cartoon, the genes are indicated as C, B, and A. In the Hfr organism, the allele is C+, B+, and A+. Usually a plus signature or plus symbol denotes a functional gene. In the F- cell, it's C-, B-, and A-, which tells us that the F- had non-functional alleles for those three genes. Now, I don't know what those three genes are, but we could imagine or speculate. Maybe the C- means can't resist ampicillin, and maybe the B- means can't resist streptomycin, and maybe the A- means can't build tryptophan. Could be any number of things. But in any case, if you knew those genes existed, but you didn't know what order they occurred within on the chromosome, you can figure it out doing something called an interrupted mating experiment, whereby you have a bunch of different test tubes, you put these strains together, and in different test tubes you stop them at different times, and you stop them by simply putting them in a blender. So let's say you incubate this strain for a certain amount of time, and what will happen is conjugation will occur. The sex pilus will form, this single strand of DNA shown here will start to spool in, and that strand will come into the recipient F- cell. Now, if you let this thing incubate for like 5 minutes, then only the A+ would make it in, and not the B or the C. And then let's say you let it incubate for 10 minutes, then you would get A and B making it in, but not the C. And let's say you let it incubate for 40 minutes, then A, B, and C will make it in. And how do you know whether they've made it in? You look and study the recombinant organisms. You look to see what their phenotypes are. And so by studying those phenotypes, you can tell which alleles recombined in this process, and you can then map or place these in a specific order. In this case, the order was A, B, and C. And this allows us to map the relative locations of genes on bacterial chromosomes, at least for those genes for which screening assays exist. All right, this is an example taken from your textbook of such a genetic mapping or mating experiment. In this example, the Hfr strain, so that's the strain that has the F factor integrated within the chromosome, which is actually a situation that you can refer to as an episome, when a plasmid can integrate into a chromosome. Anyway, this Hfr has 4 particular alleles of 4 genes. It has the AZR allele for the AZ gene, which means that it can make a protein that allows it to be resistant to the toxin azide, so that Hfr strain can grow on plates that have azide. If it wasn't AZR, then it wouldn't be able to grow on azide. It would be poisoned by it. It has the TON-R gene, which means that this particular Hfr organism is resistant to the bacteriophage T1, so it can't be killed. So you can speculate that the TON gene encodes a protein that has to do with whether or not bacteriophage T1 can actually attach to this organism, something on the cell surface. It's also LAC+ and GAL+, which means it can ferment lactose and it can ferment galactose. So it's got functional forms of all 3 of those genes. The F- is going to be just AZ, TON, LAC-, and GAL-. Okay? And so it cannot resist azide, it cannot resist bacteriophage T1, it cannot ferment lactose, and it cannot ferment galactose. So if you were interested in knowing what the order of those 4 genes are, okay, then you could mate these 2 strains together in an interrupted mating experiment. So you would put your strains in some liquid medium, and you would set them up so they would start conjugating, and then at 1-minute intervals, these are pretty labor-intensive experiments, as you might imagine, you take some of the sample out and you blend it to break apart the conjugating cells. You bust up the sex pilus. And then you plate them onto medium that contains streptomycin, which in this case kills the original donor cells as well. Okay, they're strep sensitive, so you get rid of them all by putting streptomycin in. And lacking threonine, which selects against unmated F- cells. That's sort of extra genetic details there. Okay, and so that way everything that grows is a recombinant organism only. Okay, and you grow them on 4 different kinds of media to see what they'll grow on. And then you look to see what the survival is. So if you look at this graph over here on the top right, it's the percentage of cells which with HFRH genetic markers among those recombinants. Okay, and you can see that very quickly they get azide resistance. Almost immediately after, they get resistance to bacteriophage T1, then they get the ability to grow on lactose, and then they get the ability to grow on galactose, and that there's a bit of a gap in here. Okay, so that can be interpreted as telling us that the gene order is the AZ gene is right next to the TON gene, then there's a little bit of a gap, and then there's the lactose-related gene and the galactose-related gene. And in that way we can place these on a chromosome. We can't say that there's so many nucleotides or base pairs apart, but we can talk about their relative order. And it's with experiments like this that the very first chromosome maps were actually constructed. So these were landmark types of experiments in genetics. This is an example of an E. coli chromosome map that could be constructed using methods like that. You'll note that it's given in units that add up to about 100, okay? And those are minutes. So this is an experiment done using that interrupted mating strategy. And there are many such chromosome maps that have been verified by sequencing the entire chromosome to show that the data obtained from those interrupted mating experiments is in fact correct. I mentioned this in an earlier slide, but the F factor is something called an episome, which means it's a genetic element that can exist either as a plasmid or integrated into the chromosome. There's a term here for the situation when an integrated F factor reassumes the plasmid's shape, and it's called sexduction. It's kind of an odd term. And one of the neat things that can happen here is that often when the F factor comes back out of the chromosome, and what triggers that is hard to say, but when it comes back out, it will often take along a little bit of the host chromosome or the HFR chromosome itself. And you can see that here. We've got the plasmid that's red and blue, or green, whatever color you want to consider it, but that's showing us that it's mostly F factor on that plasmid. It's a fertility plasmid, in fact, but it's also got a little bit of the chromosome stolen away from here from the HFR organism. And what's really neat with that is that when that happens, and then a routine mating process occurs, you get a situation where you actually end up with two cells that are partial diploids. They're diploid with regards to whatever genes are found here and here, which is an unusual thing in bacterial genetics and can be utilized by bacterial geneticists for certain kinds of studies. There are probably hundreds to thousands of different conjugative plasmids, and by conjugative plasmids I mean F plasmids, and many of them carry around genes that are of interest to humans because they have some sort of impact on the environment or on human health. For example, many conjugative plasmids carry genes that confer resistance to antibiotics. And so overall, it's fair to say that F plasmids or conjugative plasmids are very important vehicles of diversity in the prokaryotic cellular world. All right, so as you're organizing your study notes, I'd like you to have some examples of genes that are found on the F factor and think about what abilities those proteins can confer. So the pillin protein's the obvious example there. I want you to know what the difference is between an F-, an F+, and an HFR strain or cell. Know what an episome is and be able to provide the F factor as an example of an episome. Know what happens when an F- and an F+ mate. You should know that you get an F+ and an F+, for example. Know what happens when an HFR and an F- mate, and what you get from that is you still have the HFR cell, and then you have a recombined F- cell, most typically. And I'd also like you to have just a basic familiarity with how HFR strains can be used to map genes on bacterial chromosomes. And that concludes this part of the lecture.